kind of family you want. You want a human or bacteria family. Do you want a family you can eat and then they come alive inside of you, or that rot inside of an eggshell, right? That's, everybody's got their own preference, right? So once you do that, then the next question is, well, how do I transmit this, right? How do I get this on? If I get immune to something, am I going to pass on the immunity to you? Or to you, or to you, or to you, or to you? To my kid? Vaccine? Okay, but each person is getting vaccinated, right? Is there any way in which, and this is like the hinge of the whole debate around that, right? Is what's that? No, but if I get vaccinated or if I get resistance against something, am I going to pass it on to you? If I lick you, if I like eat dinner with you, if I Eric, your example, I understand what it's trying to do. It's not helpful for me personally. So if I put blood on you, if I like, is there anything I can do to transmit that resistance?
Mother to child, breastfeeding.
Breastfeeding? So I think in generation and in, like, I actually can't answer that. I think that, yeah, there's some level of, you're passing on genes, first of all, for sure, and then you're even passing on like chromosomal material, so epigenetic material. So yeah, there's, and then in depending if you breastfeed or not, and a lot of that will be environmental at that point, but yeah, so that's an area where yes, the answer would be some kind of a qualified yes, I think, in a lot of scenarios, right? Depending on what part of the genome we're talking about. But generally speaking, grown adults, or me and you, there's nothing I can do. I can't pass a resistance. First of all, it's not in the genome, so it's not a genetic question. It's a cellular one, which almost never will be passable, even mother to child or anything, right? There's really nothing going on there in terms of transition. So this is kind of an advantage of being the small unit, recording everything directly in the genome very, very efficiently, right? Because you are so small. So instead of, cellular, our immune system, what happens, you have a whole cascade of cells that remember it and so on, but it's so individualized, so complicated within my own body that, it's like this hugely complex fucking thing I have to unravel if I'm ever going to somehow transmit. It's never been worth it for us evolutionarily to somehow transmit, our immunity. Now, if we're in the same environment and we're having similar things happen and we find out how I became immune, well, we can repeat that. This is how inoculation starts in a lot of different folk cultures, right? So you can repeat a similar thing for my body if we think that Gigi's body is going to be kind of similar, right? And, if we've sort of, done our estimates right, it might work too. And and as we find a few, grandma died and the kid that and then we need maybe a little less, and eventually you might get it right? So this is how we get amounts for treatments. But this direct physical transfer of the actual information itself, this is the genius right here, right? So not only did they alter their genome, and they can alter this one, absolutely, right? We all can in many different ways. But having this little one means that shipping it with just connecting this P-less thing that we saw before, remember? Like, who cares about that? What is that P-less for? There you go, right? It's a little hair thing. What the fuck is it? Here, it's a super transfer physical information fucking sci-fi module, man. Look at that shit. Like, this is amazing. Put the plasmid on the diagram. Oh my God, This is like crazy, So they physically get these little bits of DNA because they're small, because they can use this little dinky, P-less freaking nothing, and it can connect with the other and then transfer over. Bam, look at that. And then they copy it, So here they got, so this is like the plasmid in action. So when a biotechnologist comes to these cells or comes to any, and they want to be doing genetic modification, they want to be mutating things, they want to create their bright red massive tomatoes, they want to, do their magic, what are they going to do? Where are they going to go? Right? They're going to go to this really easy to use small thing, and this is what we're going to spend all our time on. Now, naturally, this happens in bacteria, yeah? But now...
Why is it called plasmid instead of DNA?
There is... okay, so the word... I might get this wrong, but the word... I forget when it was coined. It was like the 20s, 30s. He was a French guy. No, it was Joshua Lederberg, who's a British guy, I think, 46, maybe. And at that time, so when we have the double helix, right? When does that happen? You guys know offhand?
Double helix?
Yeah.
I don't know what that is.
Yeah, like if I asked you in the 1920s and I said, okay, well, what is the hereditary molecule? This was the debate, right? Like, how come I got this big Jewish nose, and what happened to me, right? And then I look at my mom and I'm like, oh, okay. But then how did that happen, right? And there was a debate about it, right? And they thought a number of different things, right? They thought maybe protein, maybe this, maybe that, right? This is bound up in a lot of other things. The double helix, do you guys know when the structure of DNA, I think it's 53. Do you guys know offhand? I don't know. It's some time around there. So it could be, one reason could be, they don't really know what's on that level. And usually the name comes from the first experimental time, and often they have no idea what they're dealing with. They just have some description, oh, it's plasma-like, or it's like maybe it was in like a, I don't know. But yeah, I really don't remember, but it was a historical thing, you know, that the person who wrote the paper, where they later went back and said, this is the same thing we're talking about here and here and here. Okay, so we'll sort of keep that. So that's not going to work for you. You want to use that up there, you know, aside from it being torture to actually physically get in there. Yeah, so yeah, that's the historical name, and I don't actually know offhand what I actually read it a bit ago, but I don't remember. Anyways, you are right to say that it's tortuous to see how that's a functional, useful name for this thing here now that we're talking about, right? Like it's not apparent, and it is a historical artifact. Like so many of these names are just like either a Greek thing that they coined, or just the dude who did it, or you'll see once we get the names of like the things that cut, they're crazy, and they're all these weird acronyms of all these different historical things that aren't helpful at all. Right? They're totally not helpful. But it's like a pissing war, right? So each scientist has to get their initial in, and like, oh my God, like I did this, and I did this, and you know, and it's like, yeah, now I'm famous, you know, that one letter of like the 5 kids who read that part of the textbook, they'll be like, where did that R come from, you know? It's from Robert, man, you know. So yeah, and bio is really bad for this, because a lot of bio is kind of like, okay, here's just a bunch of stuff, good luck with it, you know, you don't really reduce things well. Now genetics and so on is getting more and more into math, but that's because we're coming with an engineering perspective, and I'll return to that. We're imposing, you know, mathematical structures in a way that we can make synthetic biology products, right? But bio itself, when we're actually describing it, yeah, it's very much a descriptive exercise. You just have this endless flow of language that you have to just somehow deal with, you know. So then what is that? So this now is a plasmid, and this is pretty much this move from this to this, which is like the last half century, is a huge move. Yeah. Once we find this, so all of that we just did in finding this and how it's working and so on, to get to this, everything you hear about genetics and the future of genetics and everything that can be done, it's not all this, right? You've heard of CRISPR. We can talk about CRISPR a little bit. This, you know, is getting back to the main genome, which is, you know, here, and then in the nucleus in our cells. We have another container for this, right? And still, the main genome, we don't have these naturally, but now we've taken these and we've engineered them, and we can start to engineer them and put them in all kinds of different hosts, we say, yeah? So we put them in all kinds of different hosts. So here we've got what you can call this sort of abstract kind of, let's bring a mathematical engineering perspective to the table. And the way that we do that is we select, right? We select, and then we reduce things, and we narrow them down, and we say, we're just going to select from this, from this, from this. It starts with saying, okay, what kind of bacteria is this? What's the most popular kind of bacteria you'll find in a lab? Does anyone know? You might know. No? No. No? Oh my God. Oh my God. She doesn't know something. Hold on. Okay. No. So what do you think? Any guesses? Any idea?
Bacteria?
What kind of bacteria? So this is true systematically. You couldn't get that wrong, because every bacteria has this, yeah, has no organelles and has this and this. But what kind of bacteria is E. coli? Yeah? So we've had like a lab strain of E. coli, a non-pathogenic strain of E. coli that basically has been the bread and butter of laboratory activity for a long time. We have what's called a model organism. Yeah? So a huge amount of research has been done on these lab strains of E. coli. You might know the Henrietta Lacks, I think Oprah Winfrey was in the adaptation. So the Henrietta Lacks story, the HeLa cells. So to model cancer, which is a kind of, they are kind of immortal cells. What gets turned off in these cells do make them kind of immortal. But those cells come from this lady, Henrietta Lacks, and it's one of the few times, again, you got HeLa cells. What are these HeLa cells? They're traced back to her first and last bits of her name. They become this, nobody thinks about it. You know, you're using HeLa cells to do all kinds of models in your lab. They're just human cells, right? somehow we've decided these are just human. Well, they're not. They're a particular person's cells, and we've mutated them in a certain way, and now we're using them as abstract models. So on the one hand, we've simplified things. We can get certain answers, but there's always the question of the black box that we're creating, right? There's always a question of, is that abstraction a good abstraction? What did we lose in that abstraction, right? What is the difference between these lab-strain E. coli and what they're doing and the real bacteria that live in the world and the real E. coli that live in the world, right? Well, one obvious thing is that they're not pathogenic. So, you know, when you lose that, you lose a lot, right? So they're different, right? So model organisms, laboratory strains. We have laboratory mice. We have laboratory frogs, laboratory fungus, and all these things, right? We have certain strains that are called model organisms that then if you look them up on a database online, you'll get tons of literature, right? And we use them over and over again because they're very useful for what they are, right? We have a ton of information. We have a ton of ability to use them. And we've sort of contained them in our own kind of space, yeah? Now, over and over and over again, this is what happens. And synthetic biology is just another layer onto this, yeah? When we bring this engineering idea, it's sort of like we've got this table, we've got different things in the fab lab. What we've done is we've taken all these natural materials and we've just layers and layers and layers of abstraction, right? Now it's the Taobao question. It's the question of ordering. It's the question of what function do I need, right? And eventually, bio is going to get to this level. I want a cell that will produce this kind of. I want a protein that does this. I want a function that does this. This kind of a question, right? Which we're in the transition of now. The same way we ask of, oh, I need a thing that does this. I need a part that fits here. I need a circuit that does this. A sensor that does this. All these sorts of questions. Now, if you try to build that thing, you want to build a thermocouple, you know, a heat sensor. You want to build a chemical sensor of formaldehyde or something. Okay, you got to go to the mountain. You got to mine. You know, you got to mine some rocks. You got to like get your little ores, and then you get them. You smelt them. You know, you heat them up. You got to have a crazy oven. You got to do all these things that we don't even think about, right? So we're going to get to a point with these sorts of things where we'll have a whole line of technologies. Right now, I can, for instance, if we go here, we can now get software, which I'll show you, and we can design this, yeah. And this right here is a software map. And then we can just get a catalog and we can order it, yeah? And not too expensive. It's gotten crazily cheaper over the years, yeah? And it's going to continue to get cheaper, right? And right now, this is a product. This is something that you can order. You can design it on software, and then you can send it to a company, and then they will give you this exact piece of DNA, right? Now, is this alive? Is it all these things? Well, no, not necessarily, but it will certainly change an organism if you did it right and you put it into something and so on, right? So the question here that we labored over, and this question of the cell and what really is important for life. Well, if we don't like philosophy and we don't like, you know, maybe it's interesting, maybe we're like, okay, who gives a crap, right? It's like we're in a forest, there's fires, there's who cares, right? We're talking about things that are changing, they're doing different things. Why is it important? Well, functionally here, whatever you think philosophically of that question, it becomes important because you have to be very sensitive to this, yeah? If you do not pay attention to this, then you're not able to do anything. You're not able to get any results. You're not able to, you maybe don't need to understand where this wood came from for this table, but you need to know something about it in order to be able to work with it, right? How thick is it? How do I cut it? How will it bend based on certain temperatures or humidity? Is it something I can have? Can I put it in that CNC machine? Can I put it in the laser cutter? You know, is it going to release some crazy chemical if I start cutting it with like a concentrated light, you know? You need to know that much, absolutely. And then your question is no longer a philosophy question, but it's a technology question. It's a question of the environment. It's a question of the impact of what we're doing. When we ask about GMOs, you know, we're putting this back in the world. Suddenly this really matters, what we're saying, you know. It might matter philosophically, you know, what the being of that, you know, GMO is. Maybe we don't care, but we're always going to care on some level ethically, right? We're going to care, well, what does it mean? If we have this much knowledge and we're putting it here, and what are we doing, right? Does it make a difference if it's on the table or if it's going in my dinner? Absolutely, right? It makes a total difference because everything around that is totally different. So what this is right here, origin of replication. This is going to be different for every organism, for every cell that I put it in. Because what I need this to do, in the bacteria, this was able to reproduce, right? It was able to reproduce, remember things, transfer that memory. How did it do it? How does that little thing, if I transfer it to another bacteria, what if that bacteria transferred one of these little plasmids directly to me? Or somehow I got it, I'm like, oh my God, the plasmid has like resistance against penicillin, you know? I don't know why I want antibiotic resistance, but you know, then it'll like kill me, right? But no, but I take it and I inject it and I get it in my cells, yeah? What will happen?
Is that something that happened?
I just did it, man. I just did it. It's done. Yeah, I just did it. I just did it. Maybe I'm crazy. Maybe there's not a lot of people out there like me, you know. But if you do it, what you're saying is, can something happen after that?
No, I mean like that is something happens in equalize, right? But like
Yeah.
human cell that
No, I'm saying, yeah, but I'm going against the grain, against Darwin, against the whole evolution. Forget it. I'm a creationist, you know. I'm just going to put it in there. I'm God, right? I'm a different kind of creationist, right? Yeah. So I'm just going to get it in there, right? And I'm gonna put it in. Can I physically do that? If that's your question, yeah, it's very easy. Yeah. You can absolutely do it. Yeah, it's very easy to get stuff into cells, and I could get it into my cell. What would happen if I got this plasmid? Yeah, I got it straight from the bacteria, straight from nature, because I'm a naturalist. Like I do GMO stuff, but I'm like eco about it. I hug trees, and then I put in plasmids in my cells, and I'm like a meditate. You know, what will happen?
Should have a sort of impact. Your recipients are going to become new donors.
Whoa. Back to philosophy. Oh my God. Did we just go back in time? Okay, so the recipients become, okay, and then you said it's going to have an effect, and then you were like going, mulling, nothing? No? Or just, I'm still not sure about GMOs? Is that not, yeah? You're skeptical that something's happening? Yeah, what?
the same mechanism doesn't exist in human cells, right?
Oh. What doesn't? Why not? Yeah?
I mean, I don't know. That's the question. Like, if it does has in like the human cell, that yeah, you will be having your like the plasma
Yeah.
or plasmid.
Okay, so this is a good point. So this is a really nice point to start with. He's saying it's not natural, so
It's not natural. It's not like saying not natural. Just like that mechanism, it happens in human body also. That basically you will have the plasmid work in your body.
yes. Okay, I'm saying it's not natural because this is what I'm taking from what you're saying and changing what you're saying. Yeah. What it makes me think of is a question of, okay, well, this is going on in bacteria. I take it, it's active here, but I put it in me, and well, it's not in any of my cells. It what will happen is one thing, but if I were to say to you then, okay, well, but I can design one of these for you, okay, for you. And you know, right now we have in Shenzhen, we had the CRISPR baby. What happened? CRISPR baby, right? He injected an embryo for AIDS resistance. I don't know if someone knows better than me. Is that what it was? AIDS resistance? So he took the CRISPR system, which was a few of these, right? Injected it in. Now that actually went to the main genome to edit it, but the plasmid technology, even with CRISPR, is still your bread and butter, okay? So the CRISPR baby, he put that in, right? And I mean, if someone, you think someone came along and said, hey, wait a second, but that's for bacteria. That's not going to do anything. That's not going to work. Why are people so scared, right? They're like, oh, dude, it's for bacteria. Oh, okay, well, yeah, we got to cancel all the news articles now, right? Like, why were we so excited, right? Why are people so freaked out? Why do they think that could have an effect?
Sorry, I didn't remember that's a CRISPR thing.
Okay, but you guys know. Hands up if you know the CRISPR baby that I'm talking about. Yeah, we know. Okay, so some people know. Okay, so sorry. So for the people who don't know, Shenzhen, about 6 months ago, maybe, there was a research scientist who injected. So CRISPR is a new gene editing technology, and I can tell you a bit more about it. But basically, like this, it's another immune system of the bacteria. It just happens to be the best one technologically. It's super precise. It's efficient. It has really good lab results, and so we've technologized it, right? This is what we do. Turn it into capitalism. Everybody's trying to brand it. Boston's going crazy in America. You know, like this is what we do. So, yeah. So CRISPR, yeah, CRISPR, yeah, CRISPR, CRISPR, yeah, and so we turn it into a technology. So it's the latest and greatest in gene editing. We've been gene editing for many decades, but this is just better. It's better. It's quicker. It can cut directly 2 strands inside your DNA. Other things could do that before, but this one's better. It's faster. It's more efficient. You get better statistical results when you run it and you see what happened, et cetera, okay? But what's at the core of it? Still this, right? We're still taking this plasmid technology, yeah? So our question now is, why is that a big threat? So what he did is he injected an embryo. and the idea is that he makes this modification, and it'll go into the genome, and it'll edit the genome. And if you do it in an embryo, we've shown in mice and so on that it'll take. Yeah. If we do it as they grow, then it becomes more complicated. Yeah. And how that happens and so on. And there's an idea in ecology, actually, that is a good analogy called a gene drive. Most GMOs you put in the environment, like any lab animal or zoo animal, just get obliterated because they're just unable to compete, right? But there's something called a gene drive where it's able to compete, and then actually you'll see the pattern is crazy. Like these things spread super fast, and in a crazy, crazy way. So a live GMO that has the capacity to spread itself, which, like I'm saying, statistically is very, very, like you really need to know what you're doing and really engineer it for that. Any GMO that anyone's doing in a lab just won't, basically speaking, won't have those qualities. So this is sort of that's the broad strokes of why an embryo, and you can imagine that when you're doing something that's still developing and stuff, okay, why it would, work better and so on. So he injects in an embryo of a kid because they did I guess they did a genetic screen. I don't know the details here, but I'm guessing they did a genetic screen. They found, oh, he's going to have this and this, or he's susceptible to this. Well, let's change it, right? So go in and we inject it, and the whole world exploded, right? So now the press within science, within the geek world, is going crazy. All the non-geeks here who haven't been following, this is what us geeks have been doing for the last half year, yeah? So as you can hear, the enough. Now, close cut. So you'll like all not out. If this is you express protein, and etc. Yeah. Me do all without crazy. They, and how do that in, was the body, right? It's not when it in there.